Okay, so this morning, this session here is for the what we call the Dataset Showcase. The idea behind this session is that so you've already looked at one dataset this week pretty closely. That's the RBC dataset that you're working on your mini projects on. And I'll say just a couple words about that in a second here. But the purpose of the session this morning is to introduce you to some other datasets that you might want to use in your Hack Week projects next week. So if you really like working with the RBC dataset and you want to keep working with it, that's totally fine. We encourage you to do that. There's a bunch of other datasets out there that are similarly easy to access and to use. And so we just kind of wanted to give you a bit of a tutorial on how to get access to some other datasets. Additionally, I'll just say up front that there's a little bit of a bias in these datasets. I work on vision and Ariel works primarily on white matter. And so the datasets we're looking at today tend to be visual or white matter adjacent datasets. That doesn't mean that those are the only datasets you should consider working with. If there's another dataset you're interested in, let us know and we can help you get access to it or help you try to get it downloaded or whatever you need for your projects. One other thing I'll say right now about the mini projects. So you'll notice that after this session, we have a break, and then we have a session with Elizabeth Dupree. And then after that, we have lunch. And then there's another hour and a half for mini projects. And what we're going to ask is that by the end of that hour and a half, by the end of the break after that, the mini project. So by the time we start the 3:30 session, everyone needs to have sent us your prediction files on Slack. So you can just send either me or Ariel a direct message with the TSV files that you've written with the predictions for your mini projects, and then we will accumulate all those and have a report on Monday of how you all did for your projects. Okay, we will post some instructions and reminders about this on Slack as well. But that way you guys will have basically from the beginning of lunch until the last session to work on your final pieces of your mini projects. Okay, so for the dataset showcase, we have if you go to the hub and you go to your home directory and then to the curriculum, there is a folder here, Rokem Benson Dataset Showcase. And this has a number of notebooks that each of which demonstrate how to access some database or some datasets. And we're just going to kind of walk through these right now. But these notebooks are here with plenty of explanation for later if you want to review or you think you want to work with one of these datasets and you want to start with some of the code we have. Okay, so I'm going to start here with this notebook 000 paths intro, and I'm going to go through this very quickly because we've already talked a little bit about this library. But essentially, all of these datasets that we're going to access, we can access with the Cloud Pathlib library that we've been using for the reproducible brain charts dataset. So this first notebook just contains an introduction to the idea of the Pathlib library with path objects. And it kind of talks through how we used to do things with the OS library, for those of you who have ever used the OS library. And now we can use this Pathlib library to have a much more kind of object-oriented interface. And basically, as we've seen before, we can make path objects to particular directories or files on the hub using this library. And there's a variety of kind of utility functions that support file system operations in this library. Okay, I'm not actually going to go through this whole notebook because we've already kind of seen Pathlib and Cloud Pathlib. That said, if you have questions about it, I would start with this notebook, which kind of demonstrates, and if it doesn't answer your questions, then come to us. Okay, so I'm going to move on now and talk about the Human Connectome project first. So you've probably all heard of the Human Connectome project. It was a great big project to scan over 1,000 individuals with 70 high-quality imaging. It was pioneered largely by the Washington University and the University of Minnesota. And in particular, we're going to be looking at the young adult human connectome here, which is the project to scan about 1,200 healthy young adult brains as sort of a way to come up with a benchmark dataset. There are other Human Connectome project add-ons. So if you're interested in things like aging or clinical disorders. There are data sets that involve that, but we're just going to focus here on the Young Adult Human Connectome Project. Okay, so the Young Adult Human Connectome Project includes about 1,200 subjects. They're all between the ages of about 20 and 35 or so. It includes high-quality structural MRI. It includes some diffusion-weighted imaging. There are tasks. There's a few tasks like working memory tasks that are part of the scan set. And then about 200 subjects participated in 7 Tesla functional scanning, where they included retina topic mapping for those of us interested in vision, as well as a few other tasks. There's also a whole bunch of twins in this data set. So if you're interested in heritability, it's an interesting data set because there are so many twins. In fact, of the 200 subjects about 180 actually subjects who did 7 Tesla scanning, I believe all but something like 10 of them have a twin in that data set. So there's a lot of, if you're interested in genetics, this is a great data set. The data have already been extensively pre-processed, which makes it a little easier for us to deal with. There's information about their pipeline here if you're interested. Okay, so accessing the Human Connectome Project, if you're following along with me in this notebook, it's not going to work with you for you unless you've already followed some instructions for getting access. So the Human Connectome Project, unlike some of the other data sets like the RBC dataset, requires that you sign like a form with them or register yourself with the Human Connectome Project. And they will give you a set of credentials that will let you access their Amazon S3 bucket. There's instructions in here on in this notebook on how to get access. It's really not very hard. It's all automated, so it's not like you have to talk to someone. You just need to submit something to to the Human Connectome Project, and then they'll give you, or you basically need to sign up for their website and agree to their terms, and then they'll give you a set of credentials. Again, there's instructions here, and if you have any trouble, we can walk you through it, but you have to do that before you get access. Okay, I've already done that on this computer, and I'll just show you here that basically the HCP is on S3, just like the RBC dataset. So we can make an S3 path object, which is one of these cloud path lib objects again. And we just point it at S3://HCP open access. And then this is a subdirectory that contains the actual subject, preprocessed subject data. And as before, we had to make an S3 client, but in this case, because we do need to sign in, we just need to tell it what credentials to use. That's what this does. So here I've made this HCP path. And if I just want to get a list of all the subjects in the project, we can just get a list of the subdirectories of the root of this project. And here it'll just print the first 10 or so of them. Excuse me, all the Human Connectome Project's subjects have 6 number IDs like these. So these are all subject directories. We can look inside of one of these directories. There's a function we have in this utilities.py file that'll just like crawl the directory and print out all the contents. So if we for example, want to look at the free surfer data for this particular subject, we can go to the base path / their subject directory / their T1 weighted preprocessed directory. And then there's another directory with their subject name that contains their free surfer data. This is all just how the Human Connectome Project organized their directories. And if we crawl that directory, those of you who are familiar with free surfer will probably recognize this file structure as being the output of running free surfer on a T1 weighted image. Okay. I'm going to go ahead. This will only take a minute or so, but I'm going to go ahead and interrupt it anyway, so we don't have to wait for it. Okay. So just to kind of emphasize that all this data is just there for you to download or use with the Cloud Path Lib. Here I've pulled out the path for a file, brain.mgz, and we can just load that in with NeBabel. Whoops. And here I'll just plot a slice out of that image, and there we have a Human Connectome Project brain slice that was just pulled down from Amazon S3. Okay. So this notebook, again, just kind of walks through how you can access the Human Connectome Project, and it includes all the instructions you need to follow to use this data set. All right, I'll go ahead and talk now about the Human Connectome Project Tesla Renatopy dataset. This is actually a dataset that Kendrick and I worked on a few years ago and published. And basically, so I mentioned that about 200 subjects, actually about 180 subjects of the Human Connectome Project participated in high quality 7T imaging. Part of that imaging was to do retinotopic mapping. Now, I'm talking about this data set partly because I know there's a lot of vision people here this year. And so if you want to look at retinotopic maps, this is a good data set to do so. There's 181 subjects in it. And what I'm going to demonstrate here is rather than accessing it using NeBabble, there's a library I maintain called NeuroPython. And NeuroPython includes a bunch of utilities for mostly reading in FreeSurfer and Human Connectome Project subject data. But here I just basically configure it a little bit to know where like the Human Connectome Project credentials are. And then I can just load one of the Human Connectome Project subjects here. And what the rest of this cell does is it makes a couple flattened representations of the cortical surface. I'll go ahead and start running that. It makes a couple flattened representations of the left and right hemisphere cortical surfaces, and then just plots them using Matplotlib. And this will take just a second because it actually needs to download a bunch of subject data in order to make these flat maps. So I'll let that run. And then there's some demonstrations of ways to make other kinds of plots here, but I'll kind of breeze past that. The data set itself, so first of all, I should back up, since not everyone thinks about vision as often as I do, and talk about just briefly what retinotomy is. So in vision, in your visual cortex, there are maps of the visual field that get laid out on the surface of your visual cortex. And retinotopic mapping is about figuring out the organization of those maps. So let's see here. Okay, so I will just go ahead and make a plot of those maps, which should make this a little bit clearer. So this is the occipital pole that we're viewing here. And the color here corresponds, well, let me make this cell here. Okay, if this is your visual field and you're fixating at the center of the screen, like this is your lower vertical meridian and your upper vertical meridian, and you see these are in red and cyan. The color here on cortex is the part of the visual field that part of cortex responds to. So this here is cyan. That part of cortex responds to this upper vertical meridian. So these kinds of maps, if you're interested in vision, these kinds of maps are available in the Human Connectome Project retinotomy datasets. They're pretty easy to access and work with using NeuroPython. If you're interested in doing anything with this dataset, you should come talk to me because I have lots of opinions about it. But this notebook demonstrates how to download and plot and deal with it. Okay, and I think next we're going to have Ariel come up and talk about the Human Connectome Project diffusion dataset. Either way.
Okay. So yes, next I'll talk about the Human Connectome Project's diffusion data. Maybe I'll preface, and at the risk of repeating ourselves, that the goal of this session is twofold. One is to give you a little bit of inspiration for next week's projects. The Human Connectome Project happens to be like a real good workhorse for hack projects because the data is readily available. It's very, very good data. They really did with the data collection, particularly for the diffusion that I know very intimately. It's really diffusion data. It's hard to find that kind of quality anywhere else. Definitely at this scale. So to kind of give you a little bit of inspiration, we're pointing you to this data set that we think is really great for things you might want to do next week. The other thing is just to show you a bit of this mode of accessing the data through the cloud that you just might not be really used to thinking about. I think mostly when we do analysis of some data sets, some publicly available data set, our first thought is like, okay, I'll download the data set to my machine, and then I'll start working. And what we'd like to encourage you to do is to skip that step of downloading the data to your machine. Of course, if there is data that you need to download into the hub in order to do things that you're interested in, we can do that. But for a lot of the data sets that you might be interested in, this kind of access is possible, particularly for Human Connectome Project. We'll show you also Open Neuro in just a minute. So just to plant that thought. So, okay, so diffusion MRI. For those of you not familiar with diffusion MRI or diffusion weighted MRI is sensitive to the microscopic motion of water at every voxel. You heard a lot about that from Serge the other day, so I don't need to say a lot, but here are just a few maps of the kinds of tissue properties that you can derive from diffusion MRI, mean diffusivity, fractional anisotropy, and the principal diffusion direction that is then used in as a cue for tractography. So here, kind of like the standard thing that we do from pathlib, import path, and then we have these utilities. Again, we're pointing to the HCP open access path, and there are a few different things there at the top. We're primarily usually interested in this most recent version, which has the 1,200 subjects. What we could do is the diffusion data is a little hidden inside there, so I'm just kind of pointing to where it is here. It's inside of, confusingly left for every subject, it's inside of a folder called T1 weighted, and inside there, there's a folder called diffusion, and that's where the diffusion data is. So for example, yeah, okay, so you can see that there are B values and B vectors, which are the metadata files that encode the diffusion directions and the diffusion magnitudes. And then there is, so here we can point to the data. I think this will go quickly because NeBabble is lazy loading the file, but this one here, the data is very, very big. They've collected 270 volumes that include 3 different diffusion weighting directions at a resolution of 1.25 millimeter isotropic for every subject. So it's very small voxels with lots of diffusion directions. And this operation here requires a lot of memory and takes a little bit of time, so I won't do it. What I will point out, though, is that though you can get the raw diffusion data directly from the Human Connectome project, sometimes what you want is something else, like you want the streamlines from tractography from the diffusion MRI dataset, or you want the tissue properties along the length of tracts. And we've made a shortcut for you by processing the Human Connectome project diffusion data through a pipeline that we developed. John, who some of you have met, is the primary developer of this software, PyFQ, that our lab develops, that takes the data, does the tractography, extracts out the major tracts, and then along the length of each tract derives tissue properties like fractional anisotropy and mean diffusivity. There's a link here. You can go see the pipeline and learn more about that. But for now, I'll just point out that if we go to this other location, this Open Neuro Data bucket, that's a different bucket. It's a project that Open Neuro Data project that's led by Joshua Vogelstein's lab at Johns Hopkins. That has a lot of other data sets, including this data set of the process data. So if you go to this particular path, you will find there for every subject, lots of different things like the regions of interest in that subject's individual space for finding the various tracts. Well, maybe I'll stop this. And then lots of results, including, importantly, tractography, whole brain tractography, but also tract profiles. So here's the tract profiles for that individual. And then you can derive out, for example, for the corticospinal tract, the fractional anisotropy along the length of the tract. So you can derive those things there. This, again, this just points to that. You can explore it a lot more, but just to point out that there's a Human Connectome Project diffusion in the Human Connectome Project bucket. And then there's this other bucket that has process data of the diffusion. So if you're interested in diffusion, but you're interested in starting from a further starting point than the raw diffusion data, this could be a good way to go. Okay. What questions do people have about diffusion, about anything so far? I see back there a question.
Out of curiosity, do you have any measure of uncertainty on those metrics?
Do I have a measure of what?
Uncertainty.
Uncertainty. The question is whether we have measures of uncertainty like error bars on this thing here. not really at the level of an individual, but we can talk about that more. I'm curious to hear what exactly you mean. So let's take that offline later. Other questions? Okay, let me go next to another thing. So another data set completely, or actually a big collection of data sets is OpenNeuro. So OpenNeuro is the Brain Initiative funded archive for human MRI data. It's actually expanded beyond that, well beyond that, into also human electrophysiology. It also includes data sets that are not human of MRI data. And this archive is pretty incredible in that it gives access to lots of data sets. You've already, I think throughout this week, you've heard about data sets that are there, and it's really easy to explore and search through. Here's one data set. This one is a data set from NYU that is a retintopy data set. So you've heard about retintopy, and we're a little retintopy centric here, but that's okay. The example will apply similarly. So there's a particular way to access the data that we've put down here. Again, this is, you can access it anonymously. It's openly available. So in this case, we put in this no sign request true in this S3 client as a way to tell Cloud Path Lib that that's the way it should access it. And so the way this bucket works is this openneuro.org at the beginning, and then an identifier of the data set. And that identifier, in order to know exactly what identifier you need to put there for the particular data set you're interested in, you will want to go to openneuro.org and search. It's very evident, like once you search, you see these identifiers in the URLs of the data sets. It's not hard to find this number. Once you put it there, you can do, you know, all the things we've shown you before. So now all the data is accessible for this particular one. There are many subjects here with retintopy. One of the nice things about OpenNeuro is that all the data sets conform to a very specific standard, which is the Brain Imaging Data Structure Standard, or BIDS. So they all follow the BIDS structure, which means that you can expect there to be a participants.tsv file at the top level of pretty much every data set there that you can then read. So for example, here's the information provided for this data set about the participants. Participant ID, just to point out, participant underscore ID is a name that's a reserved name in the BID standard. So if you're interested in doing, for example, exploration across multiple data sets on OpenNeuro, you can look for the participants.tsv file in every one of them, and then find the participant ID in every one of them, and every one of the participants.tsv. You can do, because this is those are all BIDS data sets, you can actually use BIDS in order to query the data sets. We recently did this thing where we wanted as many T1 weighted images as possible. And a student in my lab, Asa, wrote a script that basically went to every one of these data sets and queried to find all the T1 weighted. And it takes about 10 minutes to get all the T1 weighted images, the names of all the T1 weighted images on OpenNeuro, of which there are many, many T1 weighted images. So, okay, so you could get the participant names, and then you can, once you have the participant names, maybe you can say, I want for one particular subject. In this case, there are derivatives also. So BIDS allows you to attach to a data set not only the raw data, but also derivatives. In this case, FreeSurfer has been run. So you can get, using NeuroPython, you can get the FreeSurfer information for a particular subject, and then do the thing where we draw flat maps. I don't know if this will show retintopy. No, probably not. And it's going to take a minute or two. So while that takes a minute or two, a question back there.
And try to query data across dataset for legal, because
Yeah.
we have a harmonized query. Because you can go on the OpenNeuro and we eventually actually say a subfield of legal, you can search OpenNeuro plus other things, but OpenNeuro particular across dataset. We can harmonize the edge and you will say
Right.
you will speak that might be useful for you.
Yeah. I mean, as JB points out you can use NeuroBagel in order to use the harmonized. So he showed in his talk that like handedness, for example, is just a mess. If you want like everyone who's right-handed in this data set, you will have a hard time, or age or other things like that. So using NeuroBagel to query across all these data sets is also an option. This is taking a while to do. Oh, there it is. Okay, here are the flat maps. So finally you get the flat maps. So you can see, and again, just to reemphasize for you as you're thinking about projects next week, at no point here did we ever like have a step where we're like explicitly like, oh, let's copy all this data set onto my machine and then start looking for things in it. Instead, we keep as much of the data in OpenNeuro until we need it. So we'd encourage you to do that in your projects as well. Okay. Next, I will ask, so the next data set we'd like to talk about is the natural scenes data set. And because Kendrick is here and he knows so much more about natural scenes data set, ask him to talk about natural scenes data set. So maybe come up here and we'll connect your laptop.
Yeah, I heard that.
Or should we can pull the PDF from here also from your.
No, I have others.
Oh, you have other stuff.
So then maybe this can keep driving
They're gonna break.
Zoom and he can just join on the share screen.
Yeah, maybe join Zoom and share screen and then.
Then request. You gotta stop sharing.
Wow. Keep sharing.
You can keep it there. Trip over. Wait, why can't you see? Can I have share?
Wait, man, I think
Yeah?
we should, yeah.
How long do I have?
And then-
10 minutes?
Yeah.
20. Yeah, I'll kind of wing this. I'm trying to gauge like what the point of me standing up here is, as I talked about yesterday, like the point of the talk. So I got some sense. There's a lot to know about the NSD. This is something it's been completed for a couple of years now. It's a large data set. It's one of these data sets where a few people got scanned many times. This is a 7 Tesla fMRI primarily data set. So my goal, I guess, I'll just give you some very brief conceptual overview, and then get more into like concretely if you actually want to use these data, kind of the ins and outs of doing that, accessibility. As you might know, like data sets are hard if they're big. There's lots of files, lots of ways of interacting with it. And NSD in particular has a lot of, we did a lot for you in a sense, if you want to use what we did, kind of pre-processing and preparation. The raw data, of course, is available. So do whatever you want, really. Okay, so just very briefly, and please jump in if I'm not making sense or you have specific questions. Okay, very briefly, it's a visual data set. And I almost don't even want to talk about this, but let me just show you the experiment. It should appeal to people who care about vision. There's actually a memory component also buried in there. The subjects are doing a memory task as they see a bunch of stimuli. And given the scale, it appeals to computational people who want to fit models, who need lots of data points. So let me just show you the experiment. It turns out the experiment is actually very straightforward, which is nice because there's lots of other things to expend brain power on. This is the experiment. If you were a subject, you're laying in the scanner, and we have you fixate this little dot. So we want you to keep your eyes on that dot throughout this whole run. Meanwhile, these images come up. These are colorful natural scenes taken from a computer vision database. So that was intentional because the computer vision database images have annotations. So they've already labeled objects and faces and things like that. The presentation rate, 3 seconds on, 1 second off. So it's very slow for vision. Vision is very fast actually, but this is slow because MRI signals are slow. Okay, so you're sitting there, you're staring at the dot, and we're having you make a behavioral response every time an image comes up. And your task is to press, I actually forget which button is which, but you're gauging, have you seen this image in the experiment before? And it's very hard because these people are getting scanned for once a week, roughly for an hour, up to 40 times. So throughout 40 hours of scanning, you will see it turns out 10,000 different images. And each image is shown 3 times. So you're doing this kind of memory task. It's very challenging. The point was to keep people engaged, awake, and have an actual button press so we can have some measure of cognition, right? Okay, anyway, so that's the experiment. There's a lot one can do with this data set. Just to give you a brief overview of what is in it. So as I said, it's primarily fMRI data set, so the modality of fMRI, but we do have all the usual structural things, T1 weighted, T2 weighted stuff. There's some fun, you heard from John, like fMRI is actually measuring blood. So there's measures of structural vessel stuff, venogram, angiogram. I can't think of, I don't know if anyone's actually used that part of the data, but this is something I actually study, how this affects the fMRI signal. Anyway, there's a lot of different data files in there. So if this wants to be the focus of a project, great. And this slide deck, I think, is available to you. So there are some links here. So turns out preparing a data set well, I think, is a challenge. And we have lots of material to try to help you understand and wade through what's out there. So these are all links. Hopefully they're active. So I just want to give you an overview of what it is. Ariel mentioned accessibility. So once you get an idea of what to do, actually doing it is a whole different ballgame. One thing we set up, I think this is cool. This is just my browser, and you can instantly download any file on this 14 terabyte repository that you want. Obviously, you wouldn't navigate and use this for actual analysis, but this is more for manual inspection. If you just want to browse the directory tree, this is available. So that's one way you can interact with the data. There's a whole manual for the NSD, and it's like several hundred pages long. You can download the manual as a PDF, so you can search it, or you can just go to this little website. It hopefully is useful. It tells you everything you want to know about the NSD data. So there's that. And then I think this is actually very valuable if you have an hour of time and you actually care about knowing everything there is to know about the data files and how to look at them and kind of conceptually like how to think about it. There is this video, which is recorded a couple years ago. 4 years ago. I would recommend just sitting down and listening to this, and it'll show you everything. Okay. Oh, right. Ariel wanted me to talk a little bit about the types of processing, right, betas. So, okay. I'll try to explain it at some level that hopefully most people understand. So fMRI is you get a new volume every roughly 1 or 2 seconds, depending on what your TR is. So fundamentally, it's a temporal imaging method. And the raw data, of course, are need some pre-processing. So I just actually have a quick example here. Looking at data is really important, like for quality control. So this is just raw data. This was from NSD, and this is heavily sped up. There's 188 volumes in one run. So these were 5-minute runs. And I actually chose a run where you can actually see head motion. So we don't like head motion. You can see it right here. There's a little bit of jerk in the head, but these are raw data. The TR in NSD was 1.6 seconds, turns out. Anyway, so this is just inspecting one run out of the many hundreds of runs in NSD. You can already see some things. I'm a little digressing, but I can't help it. There's artifacts in fMRI data. Hopefully you understand. This is distortion and dropout, some weird combination of both. You can also assess temporal signal to noise. As you can see, it's fairly stable staring at this movie, which is good, except for these brief head motion events, like right around there. Anyway, so that's raw data, and then everyone has to pre-process it to some degree. Motion correction is at minimum necessary, because if you look at the placement of the head at the beginning versus the end, there's some shift. So it kind of doesn't even make sense to analyze it unless you at least try to resample the data to account for that shift. So at minimum, motion correction. So that's like arguably the first standard step. And so this is the same. This is 4.10.6. So this is the exact same data after what we did to pre-process it. As I mentioned, we did a lot, and you could do it yourself if you want. But this is the exact same data And so if we scrub through this movie, it's a lot more stable. But there is still badness, right? That head motion event irreparably corrupts the data. So you can actually still see there's some strange looking thing that happens around here. Anyway, so that's, minimal pre-processing. It's often referred to as, motion correction. And there's also spatial undistortion stuff for those who understand about fMRI. Okay, and then at this point, the data you can think of as time series. So if you query one voxel, your favorite voxel, you can look at the fMRI time series. After that, we do more because it's still quite large and arguably not as useful. So in NSD, which you can, oh, I forgot to mention, there's the paper that describes all this. There's a lot of information here, but anyway, so given the preprocessed time series, we furthermore analyze it with a GLM, so a regression model. In this case, we used a single trial GLM, we call it. I think it's pretty standard terminology. The idea being we want to estimate the amplitude of the bold response for every single trial. And in NSD, a trial is you get an image for 3 seconds and there's a slight gap. So we already applied, using various fancy techniques, some method to extract single trial bold response estimates to every single trial in every run for every subject. So there's that version of the data, which is further processed. And for the most part, I assume almost everyone who's used this data set. Oh, and I should say, like, this data set has gotten a lot of uptake, primarily in like the computational community. I assume most people or everyone uses the betas because there's a lot of data, and just loading the data is very hard, actually. So if you work with the betas, this is just a figure from the paper, just to give you a sense of what I'm talking about. This is a big data matrix where the rows are different voxels taken from some region in the brain, and the columns are individual betas, so bold response amplitude estimates on individual trials. And this is taking the data across, in this case, 40 scan sessions from this subject. So if you zoom in on one of the sessions, session 10 here, I guess you can maybe see more clearly the data. And yeah, one tiny detail is we actually provide multiple versions of these single trial betas, demonstrating the kind of advanced techniques that we're using to try to get better and better estimates of single trial responses. The reason being fMRI is extremely noisy and you have to do something. Anyway, so there's the beta version, or the single trial beta version of the NSD, which is probably most convenient to deal with if you care about using these data. What else? There's our, so typical in vision, we have these localizers, so find V1, V2, V3 using like a retinotopic scan, which we did do on these subjects. We also have very conventional category selective localizers like for face regions and body regions. So that's all in there. Yeah, can you think of any other things I should briefly touch on? Any questions about the data set? And oh, and I'm here through this afternoon, so if you want to just chat about what this is or have an idea, I'm happy to discuss it with you. Otherwise, but yeah, I'm here until maybe 4. Okay, sounds good.
Maybe I can.
Okay, Zoom, switching computers.
So that being all said about so there is a notebook in here in the data showcase curriculum folder that also shows so same idea, different S3 bucket. This S3 bucket, Kendrick showed you a web view of this S3 bucket, where you browse the folders and files and find individual files. You can also see this S3 bucket
the cloud pathway API. So this base path, here's kind of the top level list of top level folders. So you can see there's raw data, M series, diffusion, and so on. So you look at the free surfer, you see there are free surfer folders for every subject. Another option is to go. What's PP data?
Pre-processed.
Pre-processed data at some resolution, and there you go. Files and so on, lots of files, lots of data here. For example, population of subject fields, you can get the coefficient of determination. Look at the coefficient. Again, to explore the very vast repository from where all the files are, and then find the particular files that you want, and then
We can't really hear via Zoom.
People online are having a bunch of trouble.
Oh,
Now it looks like
to hear us.
oh, are you plugged into the owl? Are you plugged into the owl? Sorry, online people.
Can you hear me now? Folks
Yes.
online?
Yes, much better.
Yeah, I'll give you a shortened version of the last few minutes, which is that there's a notebook here that shows you how to access the data that Kendrick was talking about. It's in this bucket, and it's enormous. So explore away, and then you can read data like I show here. I show a very particular example. You can read the data directly. So mitigating the need to like download and then read. Okay. What other questions do people have about NSD? Okay, one last data set here for completeness is the Healthy Brain Network data set. The Healthy Brain Network data set, you can think of it as a precursor maybe of the RBC data set that you have been looking at in your group projects. It's a large data set, developmental data set. It's eventually we'll have, I think they've settled on eventually having about 5,000 subjects in this data set, ages 5 to 21. They're collected at 4 different sites, so 4 different scanners, which makes for interesting quirks. And it's in a sense enriched in diagnosis of mental health conditions because the recruitment is done through doctors' offices. So this is so for people interested in mental health, this data set is quite interesting. Unfortunately, the kind of individual diagnoses and lots of questionnaires that included in this are actually not available unless you get a data use agreement. So that will be something that you can't do next week. But there is age, sex, handedness. There are, if you're interested in doing something further with this, there are a lot of additional phenotypic data like clinician administered case ads and diagnostic assessments for a variety of different disorders and other psychometric and health kinds of assessments. There's also, in addition to MRI data, there's also EEG data on those same subjects, making for potential interesting combinations. One particular thing I'd like to, again, pause on something that's not available yet for RBC, but is available for this data set is diffusion data. And one thing that we've done with this diffusion data is to pre-process all the diffusion data and again, run it through those pipelines for PyFQ. A kind of interesting side part of this is we spend a lot of time on quality control for this data. Kids tend to move in the scanner, so data quality can be an issue. especially for longer acquisitions like diffusion acquisitions. And one of the things that we did in order to do quality control is that we created a community science app where people could go online, see bits of the data and classify that as good quality or bad quality. And then we went through a whole process to train a neural network based on those inputs so that we would get quality assessments for the full data set, which is very large. It would be very, very hard to look through every single scan for every subject here. So this is potentially also interesting if you're interested in quality control, because we have now several different levels of quality control. There's expert quality control for about 200 subjects, or for 200 subjects from several different expert raters. There is community quality control from lots of community members for about, for 1,653 subjects. And then there's neural network quality control. So if you're interested in kind of thinking about quality control for these kind of data, this is a neat data set to use. The data set's all navigable through a similar browser to what Kendrick showed through this link. And then, of course, accessible in S3. Now in this bucket, so I should just point out this bucket FCP-ND also has many other data sets. Some of you have seen the Abide data set when I was talking about that data set during the machine learning session. The Abide data set also lives in here, together with several other data sets. So FCP-ND is another place where you can get lots of different data sets. And then you can get who are the participants, and here you have expert quality control score if that exists, but then other levels of quality control scores in that participants table, and then the track profiles. Here's one subject's track profile for the corticospinal tract. So if you're interested in diffusion, tissue properties of tracts, and so on, that's accessible in there as well. Okay, what questions do people have about this? Yes.
It's kind of impression. It's wonderful you give us this tour of the data set. And so is it always the case? Normally when there's a data set, people need a tour guide. Tour guide is difficult. You cannot, let's say you go to a website, or first of all, somebody needs to tell you that it's this data set, you need to go to this website.
Yeah.
But let's say this people already have. Then usually you still need a tour guide.
That's a really great question. So this is, I think, a little bit of a meta question about this. The question is whether we've given you a tour of several different data sets, and the question, do you always need a tour guide in accessing these data sets? And I'll say, for example, for this data set, I know that people have used this data set without me giving them this tour. There's a paper. So, and for NSD, the same thing. I think there is a paper and there's a lot of documentation. So having a tour, having Kendrick give you a tour and us give you a tour is a bit of a shortcut, but it is definitely possible to access that data. I think Kendrick can probably tell you there's probably dozens of papers written by people that Kendrick hasn't like directly given them a tour at this point on NSD. So it's definitely possible. I'll use this opportunity just to point out that one of the goals of the brain imaging data structure standard is exactly to avoid the need for, so data standards in general, the goal of those data standards is that every data set looks the same in some way. That you can approach a data set on OpenNeuro, for example. You can go to a data set and you wouldn't need a tour guide to tell you where to find the structural scans for every individual, or where to find the functional scans, or what it means, like how do I know what the subject's handedness is, or what their age is, because they conform with the standard, and those bits of information should, at least in principle, be always in the same place, or described in a way that is automatically accessible in the same way.
One just needs to be educated about the standards.
Exactly.
So where you give us also files showing go there. That's where the standard. So there is it. There's some place. So
Yeah, let me try this.
It's neural image.
Yeah, so we haven't actually taught I'm happy to do early next week a whole session on bids and explain it, because I think it's really important. Who here has let's say before this course heard about bids, just out of curiosity? So a lot of you. Who here uses bids on a daily basis? Okay, several of you as well. Okay, who'd be interested in like a session on here's bids, here's the logic, and here's how you use it, just like that. Okay, we can do one on Monday, even afternoon or something. Just we'll do like 45 minutes, so it doesn't take too much from the hacking. Yeah, I think this is really important, and if we had 2 weeks for lectures and another week for hacking, we would probably include a lecture on this. But I can give a short lecture on this next week and show you some of the tools. One of the nice things about the standard is that it just to say here is that it's explicitly meant to be both human readable, so a human can look at one of these data sets and understand where things are, but also machine readable. So there's software that automatically queries the data, and you can build really sophisticated, like I said, you can build tools that, for example, run through all the data sets on OpenNeuro and find all the data of a certain type within that. So the standard really supports a variety of use cases, and should make it, at least should make the need for a tour obsolete. Okay. What other questions do people have? Do people have in mind like other data sets that they might be interested in approaching next week? Maybe I'll take the opportunity, since we do have a few minutes here, to also talk a little bit about next week. Some of you might be wondering what that might look like. So next week is a lot more open-ended. We've already sort of shown you the schedule. It's just a lot more empty. And so what we'll do on Monday morning at 9 a.m. is we'll gather here, and what we'll ask you to do then is to come up with ideas for things that you would like to do next week. And it can be really anything that you would like to do, to spend the week doing. And then if you have an idea, you can already start pitching that idea in the Hackstorming Slack channel. I've seen at least one show up there. And it can be, I'm thinking about this data set, or I'm thinking about this kind of analysis, or I'm thinking about this software, or I would like to write a poem about this particular Norse god. It could be really anything, and just start brainstorming. But on Monday, what we'll ask you to do is to come and pitch that idea. So we'll stand up here, and you will describe that idea in just a few sentences. You don't have to prepare slides, or you don't have to prepare a very long spiel. It just needs to be a few sentences so that others can hear what you're interested in. And then on Monday, we'll also show you how, once you've done that, how you arrange yourself into a Slack channel with your group members, and also a GitHub repo. So we'll have like a template that you can, similar to the template for the group projects, the template that you can get you started to start developing your project, presumably. And even if you're writing a poem, you can put it on GitHub, but presumably most of your project will be on computational things that we've shown you throughout this week, and their Git and GitHub will be useful in order to develop that together with your collaborators. What questions do people have about that? Yes.
So additional, this is nice to know that we can go there and understand. Is there some kind of a yellow page of all databases?
Oh, like a data catalog.
Yeah, so that,
Yeah.
you know, because you are guiding us to a URL link. So is there kind of a yellow page of URL link? I mean, this is the database of or the black people, this database or the
Yeah.
you know.
So NeuroBagel that JB talked about, it to some extent you could think of it as like a data catalog, sort of, but not quite, because it's
Yeah.
okay.
Focus on you access sample.
Yeah, so you have these data sets, and you can create pseudo samples out of the data sets. JB is correcting me that I'm off. Yeah.
But it has another data set, it has OpenNeuro MRIs, plus some of the EEGs, plus some of the other things. So it's not a bad trust code.
Yeah.
There are, I think, out there, I remember at least one paper by NG or there are out there more like high level description of data set.
Yeah.
It's a different kind of a need.
Oh, so it does have some kind of a high level description of all kind of useful data set that people like to.
Yeah, I don't think there's like the one true data catalog, unfortunately. I guess Nitric, NITRC is another place where that's neuroinformatics, something resource center. I forget what all the letters there stand for, but Nitric is a good place also. NITRC, which is a website that's existed. It's been around for quite a while already and aggregates both. There's both software and also data sets there. In fact, then some of the data sets are directly accessible through the website as kind of a portal to the thing.
Technology Innovation Center, so maybe it's a.
Okay, I'll find it and I'll send it on Slack or somebody else will send it on Slack. Nitric is a good place to search. Open Neuro is obviously a good place to search, but that's just a particular slice. Yeah, I mean, potentially a hack project for next week. Create the ultimate data catalog of open data sets in neuroscience. Just an idea. What other questions do people have? Data sets next week? Okay, well, if no questions, then I think we can wrap up here now, and then we'll be back here in just over 30 minutes. Yes, question.
Think certain data bits will be easier to like use in terms of like pre-processing.
Yeah, the question is if there's like data sets that are easier to use in terms of their pre-processing and so on. So we were kind of in our presentation a little bit human connectome project centric because that data set's like it's there, it's accessible, it's pretty high quality. So if your interest is, I want some data, I want to know that it kind of things will work with this data. That one's pretty good, I think, Human Connectome Project. The challenge with the Human Connectome Project is that all the acquisitions are pretty high resolution. So the data files themselves are pretty big. RBC is very nice, actually, honestly, and is a lot less clean because it's got kids. So it's also, I think RBC is a really good, that's the reason we did the group projects with RBC. Also, all the data is accessible, multiple levels of processing. So you can do a lot of things with it. Yeah. Okay, at 10:30, we will have a Zoom lecture. So we'll have Elizabeth Dupree zooming in to give a lecture about machine learning in neuroimaging. So see you in about 30 minutes.